In Chapter 18, we're going to be taking a look at some of the mechanisms associated with the control of the bacterial genome. Bacteria tend to live in relatively unstable environments and therefore must adapt rapidly to change. For example, bacteria tend to activate certain metabolic pathways in order to utilize a resource when it is present, but deactivate these same pathways when the resource is absent in order to conserve energy and cellular resources. The main question that we want to answer is exactly how are prokaryotes able to adjust their metabolism so quickly in order to respond to these rapid environmental changes. To illustrate, let's take a look at how E. coli activate genes involved in lactose metabolism only when lactose is present in their environment. Common in the gut of mammals, such as cattle, E. coli bacteria don't normally encounter the sugar lactose. Therefore, it is expected that the genes that code for the enzymes that metabolize this sugar are suppressed in order to conserve energy and cellular resources. The question then becomes, how will these same cells respond when introduced into the gut of a nursing calf? In the gut of a nursing calf, E. coli bacteria do encounter lactose. Therefore, genes for lactose metabolism are activated so that these cells can utilize this new energy source. But how exactly are prokaryotes able to respond so quickly to these changing environments? Bacterial cells are able to rapidly shift gears in a metabolic sense due to the unique organization of their genomes. In the bacterial genome, genes that code for enzymes that are active during the same metabolic pathway are organized into clusters known as operons. A typical bacterial operon will consist of the following components. The structural genes of the operon are those which code for the enzymes that are active in a given metabolic pathway. The promoter region is instrumental in the transcription of the structural genes. A regulatory sequence, known as an operator, can be regarded as an off switch, which, when stimulated by the product of a regulatory gene found upstream from the promoter, will suppress the expression of the structural genes. In contrast with the organization of the bacterial genome, related genes for a metabolic pathway are not clustered in eukaryotes. In addition, each gene is serviced by a single promoter. The two operons that we'll be taking a look at in this chapter are the Lac and the Trip operon. The Lac operon functions in the metabolism of the sugar lactose, whereas the Trip operon functions in the synthesis of the amino acid tryptophan. Here we can see the organization of the Lac operon. Like other operons, it is associated with a regulatory gene, an additional regulatory site, a promoter, an operator, and a host of structural genes. With respect to the Lac operon, the structural genes are called LAC-Z, LAC-Y, and LAC-A. The LAC-Z gene codes for an enzyme known as beta-galactosidase. This enzyme basically breaks down lactose into the simple sugars glucose and galactose. The LAC-Y gene codes for an enzyme known as lactose permease, which facilitates the transport of lactose into the cell. LAC-A codes for an enzyme known as thiogalactoside transacetylase, whose function is currently unknown. We can investigate how the Lac Operon is specifically regulated. We have to consider some of the raw materials that may be available to E. coli. For one, the simple sugar glucose may be utilized. This raw material, because of its simple structure, does not require any modification in order for the cell to utilize it for the purposes of cell respiration. In contrast, lactose, being a disaccharide, is more complex and therefore does require an energy investment in order for it to be broken down. As a result, lactose is usually only consumed by the cell in the absence of glucose. So it's important to remember that the Lac Operon is generally only activated when lactose is present, but glucose is absent. Let's consider the regulation of the Lac Operon under the conditions in which lactose is absent and glucose is present. Under these conditions, the regulatory gene found upstream from the operon is transcribed into messenger RNA, which is ultimately translated into a regulatory protein called the Lac repressor. This protein can associate with the operator. This has the effect of blocking the association of RNA polymerase with the operon's promoter. This in turn prevents the transcription of the LAC-Z, LAC-Y, and LAC-A genes. Consequently, the aforementioned proteins instrumental in lactose metabolism are not expressed. Under the conditions in which both lactose and glucose are present, lactose is converted into allolactose, an isomer of lactose, which then can engage with the repressor protein, causing it to disengage from the operator. Despite the loss of the repressor protein from the operator, RNA polymerase does not strongly interact with the PLAC promoter. Therefore, there is not much transcription of the LAC-Z, LAC-Y, and LAC-A genes. This is because the PLAC promoter is regarded as weak because it does not have a very high affinity for RNA polymerase, at least when glucose is still present in the surrounding environment. When cells have finished utilizing all of the available glucose in their environment, levels of cyclic AMP begin to rise. This molecule can begin to interact with a regulatory protein known as CRP. The activated complex can then engage with a regulatory site found between the PLAC promoter and the upstream regulatory gene. The interaction with this complex, with the CRP binding site, increases the PLAC promoter's affinity for RNA polymerase. As a result, the LAC-Z, LAC-A, and LAC-Y genes will begin to be transcribed at a much higher rate in order to now take advantage of the lactose, the only raw material now present in the environment. It will now be apparent that the Lac Operon is an example of an inducible system in which the operon in this case is always regarded as being off unless lactose is present and glucose is absent. Now be apparent that the Lac Operon is an example of an inducible system in which the operon in this case is always regarded as being off unless lactose is present and glucose is absent. As time goes on and more tryptophan is synthesized, it will begin to engage with the TRP repressor, activating it. As a result, tryptophan is often regarded as a co-repressor. This active complex will then engage with the operator of the TRP operon, blocking RNA polymerase's association with the p-TRP promoter. As a result, the structural genes responsible for the synthesis of tryptophan synthase will not be transcribed. It can be seen that the TRP operon is regulated by negative feedback, in which high levels of tryptophan can actually end up suppressing the expression of the structural genes, therefore blocking the synthesis of the enzyme complex tryptophan synthase. The TRP operon can be regarded as a repressible system, which is always on unless a particular metabolite exists in a high enough concentration to suppress its own synthesis. This is done in an effort to conserve energy and cellular resources.